Hi, everybody. Welcome back. Today, we're going to talk about how species turn on and turn off their genes. More importantly, when do they turn on and turn off those genes? To begin with, we're going to start with the easiest examples, and those are the prokaryotes. Bacteria only have one ring of DNA, and here's a little cartoon version of what it might look like on the right. They have about 4,000 genes, 4 billion base pairs. So this is a really quick representation of what might be there. Anytime you see something expressed as KB, KBs, that's a bad K, but KBs equal 1,000 bases, and then you have MBs, and those are million bases. So I'm looking for, so at about the 1,604 kilobyte or kilobase distance from zero, that's how many genes are in there. Those genes are not all going to be on at the same time. It wouldn't make sense energetically. It wouldn't make sense resource-wise. It wouldn't make sense to have space to put all the products either. So there's going to be times certain genes get turned on, and there's going to be a lot of times that genes spend off. Humans, well, we have more than one chromosome. Humans have 46 chromosomes, 23 from each parent. I'm going to leave a link to this website. It's really, really stinking cool. Each of the 46, well, 23 pairs are shown, and in the little viewer, they go through and show you the location of all of the diseases that we have. A misinterpretation of this is that those are where the diseases are. A better interpretation is that's where the good genes are. And if the good genes go bad, that's the disease that you have. So right at the top, if you have a bad gene up here at this loci, it's going to be for cataracts. Down here, we have a gene for an opioid receptor. We have P53 genes on chromosome number 1 for regulating DNA repair before we get into the S phase, out of G1 into S. Highly suggest getting there. Our genes are scattered all over our genome. Chromosome pair number 1 for eukaryotes is not exclusively for digestive system, and chromosome pair number 3 is not exclusively for digestive system. The systems are scattered all over our genome. And so it's really important so that when you're inside, say, an epithelial cell inside your mouth, that you have mouth epithelial genes turned on. And if you're on your eye and you're talking about the lens proteins that are being made, those cells need to be making eye lens proteins and not toenail genes, right? So those are really, really, really important distinctions. And how does it do that? How does it even know to do that? Every cell in your body has all of the information on how to make a you. I've got genes on how to make a pancreas, how to make a liver, how to make feet, how to make a nose. All of it is inside all of the cells in my body, right there in my finger. But only little finger surface epidermis cells are turned on right there, right now. How is that? If you really stop and think about it, it's incredible. It's much the way a computer operates, right? So we have chromosomes, they have folders. We have genes, they have programs. We have base letters, they have little bits of information, the ones and the zeros. It's really important that when you turn on a program, certain information is only read, right? So right now I'm using Chrome, I'm using my camera and microphone, and those are the things that are operating the most. But what you're going to find too, and this might be down the road a little bit, is the genes that are going to start making epithelial cells, turn on for epithelial cells, about 50% of those are the same for liver genes. About 50% of those genes that make pancreases are the same, and gallbladders and intestines and neurons, because they all need a cell membrane. They all need receptor proteins. They all need channel proteins. They all need certain enzymes inside to carry out glycolysis and to carry out other pathways. So the beginning parts are the same, and I can compare that to a computer as well. If I'm using that little arrow, how often does that arrow show up? Think about a PowerPoint when it turns on. Think about Microsoft Word when it turns on. Think about an Excel program when it turns on. Any program, many of them just use that little arrow that comes up. So there's redundancies built into a lot of the programs we use. When you're in a Word document or you're in a let's use a Google Doc or Google Slide, your cursor blinks to let you know where it is. That's a redundancy in program. You go into Google, a Google search, it does the same thing. So even though you have 3 or 4 different programs, there's redundancies built in for the same type of program. You would find the same type of writing in each of those different programs, just like you'd find similar writings to start off with in different tissues that we have. Well, prokaryotes are the good place to start for regulation because they have a pretty easy system when it comes down to it. When we're going to talk relatively to other regulatory systems, it's easy. But it's not easy if you're just getting started at it. They use basically on and off switches. So the left-hand side, those are called inducible operons. And the example we're going to use is the operon that gets turned on to digest lactose. On the right-hand side, we have the trip operon for tryptophan. The trip operon usually is on, but then if enough tryptophan gets made, it turns off. And when you don't have enough tryptophan anymore, it turns back on. So when I say you, I mean bacteria. We don't have operons. And that's the way it regulates. Inducible operons like lactose isn't always there, so you wouldn't want the gene on all the time. So lactose, when it's present, is going to turn on the gene. Let's get started with that. To begin to understand how all of this is going to operate, we have to go all the way back to what we called feedback inhibition. Feedback inhibition. Meaning, the products, once they're made, go back to the beginning and inhibit its own production. Why? So that you don't make too much. This is chapter 8, where we have a precursor, and that precursor passes through enzyme 1, and it changes it. And then it passes through enzyme 2, and it changes it. And it passes through enzyme 3, and we get to something called tryptophan. Tryptophan, once it's made, goes back and turns off enzyme 1. It allosterically, remember that word? Allosteric. Allosteric means that on the back side of it, it had, and this is like the active site, it had a space where tryptophan could dock and then maybe change its active site enough so that it wasn't active anymore. The products also go back and they inhibit the genes. Not only do they go back and turn off the enzyme, but they go back and turn off the genes. And then once they're, and so ever so slightly, this holds on, it has what we call affinity, but it'll fall back off, right? And when it falls off, it'll go back into its configuration for its positive configuration. And then if there's a lot of tryptophan still there, it'll scoot itself in and then deactivate it. But the less tryptophan you have, the less active that enzyme would be. So you get into this homeostatic graph of if you have too much, the gene and the enzyme get inhibited so that it goes back down. But then now we're having too little, so it goes back up. And here's homeostasis. This is my high set point, and this is my low set point. And in the middle is where I want to keep things about on average. Prokaryotes, like E. coli, they live inside our gut. From the end of your small intestine to your anus, you've got nothing but undigested food, some water that you're taking back into your body, and you're loaded with E. coli. E. coli outnumber our body cells about 10 to 1 in this space. There's just so many of them. And they have their own metabolism going on. They're getting little shreds of food that pass by. Ideally, these guys want glucose. That's the best of the best, but it's not always available. Sometimes they have to settle for lactose. Now, lactose is really, it's a galactose molecule bonded with a glucose molecule, and that's lactose. There has to be an enzyme to cleave that so we can go down this pathway. In fact, this is the pathway on the left-hand side, and you'll notice that once glucose gets separated, it goes into glycolysis, and you know glycolysis. You just took a test on hexokinase, and hexokinase goes in then phosphofructokinase, and then you got your isomerase. Then it goes on until you turn your glucose eventually into 2-pyruvates. Look what you need, though. You need this enzyme called beta-galactosidase. Beta-galactosidase is the enzyme that's going to work on that. Some of the galactose is going to go this way, and the glucose is going to go this way, and beta-galactose has its own pathway to become energy. But you got to have that enzyme to start with. So this is the way it starts. Going along the DNA of the prokaryote, there's about 4,000 genes. This is the way the first one we're going to study is set up. There is what we call the repressor gene, or the regulatory gene. And what it produces is something called the regulator, all right, or the repressor. I'm going to write repressor right here. To the right of that, you have what's called the operon. And the operon is between the brackets that I made. You have the operator, right? We called that the promoter in eukaryotes. You have the operator. And then just to the right of the operator, you have a succession of genes. You have LAC-Z, LAC-Y, and LAC-A. In us, I would have called those exons, right? But prokaryotes don't have introns and exons. They're just not that sophisticated. Instead, they have the package of genes that they need in these little sections. So let's get started. This little guy, let me erase this. That regulatory gene codes for the repressor. And that repressor will rest over the operator when there's no lactose in abundance. Why? Because you don't need to do anything if you don't need to do anything, right? So all of these genes are meant to break down lactose. And if you don't have lactose, don't need to make the gene. That means that your RNA polymerase cannot slide down and read any of the genes. It's blocked by that repressor. Well, as luck would have it, our bacteria has just come in contact with a little bit of lactose. Lactose can permeate through the membrane, but it doesn't do so readily. Lactose is a pretty big, bulky molecule, so it's much bigger than glucose. Let's just leave it like that. So once lactose gets in, its shape, and I've simplified the shapes, is just the right shape for the back of the repressor. And when it binds to it, it does so allosterically, and it unlocks it from the operator. This is a key step. The molecule, lactose, is available now. Now's a good time to start reading the genes so we can break that bond. So the promoter is open, and RNA polymerase can run just like it does in eukaryotes. It reads the DNA in a 3 to 5 direction, and it makes RNA in a 5 to 3, right? So each of those genes get read. Now, those genes are going to hustle off to a ribosome. First gene is going to make that protein called beta-galactosidase. That's the one that's going to be responsible for breaking glucose away from galactose of the lactose molecule. The second gene that gets read creates something called permease. Permease is a channel protein, and it's going to rise up to the membrane, and it's going to act as a funnel to let even more lactose in. And then this last molecule called transacetylase. Transacetylase is sort of a garbage disposer. Once this metabolism starts taking place of breaking apart lactose into galactose and glucose, it's going to have some toxic or not wanting to keep around byproducts. So that is going to detoxify them and kick them out of the cell and not let them back in. So here we have our membrane. Here we have one of the permease molecules that is now letting in more lactose. The more lactose that comes in, the more lactose binds to that repressor, and the more repressor is kept off of the operator. Every now and again, a lactose will fall off the repressor, but since there's so much lactose coming in, another one will scoot over and take its place and keep that repressor off. So that allows RNA polymerase to keep sliding through and making more enzyme, making more permease, making more trash destroyers. And it's sort of a positive feedback. And this thing keeps cycling up and cycling up. Well, eventually, it's going to digest some of the lactose. And as the enzymes get used up and they're not really there anymore to break down lactose, they'll be degraded as well. And eventually, once all of the lactose that came in has been metabolized, we just go back to the way things were. The repressor goes right back onto the operator. The RNA polymerase goes back and it just can't read the rest of the RNA. And so you've effectively turned on the gene for the time that you needed it. And now that we don't need it anymore, we've shut it off. You're going to save on energy. You're going to save on resources because every time every one of these genes is read, right, transcribed, they have to go over here and they have to make the enzymes, which means you're going to have to take amino acids out of the cell, out of the bacteria, and put them into making enzymes. That would be a waste of materials. So it's conserving. It's fascinating. All right. This is the book's rendition of it. It's the same thing. You have a long promoter region. They put in the operator. I just called it a promoter so we could start, but it is the operator, just like in eukaryotes. And the active repressor, beta-galactidase, goes in there and it will bind with the protein. This is what it looks like when it's active, right? It'll crank out all of those enzymes and the protein that goes in the membrane and the little enzyme that's going to be our digester. When all is said and done, it goes back to being off. So the thing to remember about the lac operon is that they start off. They have to be induced to start. That's why it's called an inducible operon. Starts off, it has to be, I like that word, coaxed. It has to be coaxed into believing that it can start its day off and start breaking down some galactose. Well, it stays active until your inducers are eliminated. And the lac operon is really the textbook example of what we call an inducible operon in bacteria. It's the one that's the most studied. Okay. So now the other side of that is the repressible operon. In repressible operons, these little guys start on. So I'm going to jump ahead. I'm not going to use the same cartoons I did in the beginning. I'm going to use the books just to start us off because you have a better understanding now. You still have your promoter region. You still have your operator. Every time I hear the word operator, I think of the Matrix. Operator. RNA polymerase is going to slide down, and as it does, it's going to make a series of enzymes. It's going to make enzyme E. It's going to translate into enzyme D, C, B, and A. All of these are enzymes, and all of these arrows are shorthand for translation, right? Because this part is transcription. All right. Each of these enzymes is going to take a precursor that the bacteria has absorbed. And then enzyme 1 is going to work on it, E, and it's going to change its shape. And then D is going to work on it, and it's going to change its shape again. And this continues and continues until enzyme A is your final enzyme, and it makes this little amino acid called tryptophan. Tryptophan is abbreviated as, TRP. And you've noticed this, you've used tryptophan in your amino acid chart. Tryptophan is a crucial amino acid used in a lot of different proteins that the prokaryote needs. So it needs to be synthesized. And as it's being made, it's going to be made in abundance. It becomes something called, remember this vocabulary term, a co-repressor. We already learned about repressors. The trip operon has its regulatory gene, and it makes its own repressor. But I want you to notice something about shapes right now. If the operator has that shape, this is not the right shape. These little guys are pointing off at about 15 degrees off in the other direction. It's not exactly the way it works, but it serves a purpose right now. So what's going to happen, I'm going to clear all this off. What's going to happen is that tryptophan, once it's in abundance, starts to bind to the repressor. So here's my repressor, and here is my tryptophan, and the tryptophan becomes something called the co-repressor, and it has an allosteric site that it binds to, and when it does, it changes the repressor shape into a shape that then fits with the operator. Why? You're starting to make enough tryptophan. If you have enough tryptophan already made that you have extra to fill the pocket of a repressor, that means that you have enough. It doesn't stay there all the time, doesn't stick. It has an affinity. It'll hold on to it for a few seconds and let go. But once it's a hold of that repressor will slide up into the operator, and now RNA polymerase can't read the gene anymore. Since it's going to be there and it can't read the gene, that means that the tryptophan is going to start getting used up, and eventually it will be used up so much that there won't be enough tryptophan left over to hold on to the repressor. It'll fall off, and then we'll go back to the gene being read again, and then we follow the cycle through again. If I were to graph the amount of tryptophan there is in a bacterial cell throughout the day, it would probably look something like this. I have highs and lows. We call that a homeostatic graph. You have high points, you have your low set points. It just falls in place. This is the way they regulate a different way. With the lac operon, they only turned it on if they need it, when something showed up. Now you always need tryptophan. And since you always need tryptophan, you don't want to exhaust yourself making the tryptophan. So we're only going to make enough that we need. How do you know you're going to make enough that you need? Because we're going to make enough for the biopathways, but we're going to make a little extra that can grab a hold of that repressor and turn off the gene when we need to. And if there isn't enough to turn off the gene anymore, then the gene gets turned back on and we can start metabolism again. So the thing to remember about repressible operons, my picture went a little far, there we go, is that they always start on. They're always on. They're only turned off when the products are in such abundance that they come back as a co-repressor and grab a hold of the operator and turn it off. Stays inactive until your trip decreases, and this is going to be the textbook example of a repressible operon. All right? We're going to cover eukaryotes next. So, lac operon. Lac operons are inducible. They start off. They have to be turned on. The trip operon starts on, but it eventually gets turned off. Both are regulatory ways inside a bacterial cell to control enzyme production and production of metabolism. On one side is going to be galactose and lactose. On the other side, it's going to be an amino acid that you need for normal metabolic activity. Okay? I hope this video helped. I'm going to throw a couple extra little ones in there for you to check out. And if you have any questions, you know how to email me. I should open up comments. You can throw stuff in the comments too. Thank you very much. If you like what you heard, don't forget to click subscribe and like or follow or whatever it is they say, because I have no intentions of monetizing this, but it's always good to hear feedback. Feedback, get it? All right, guys. Talk to you soon.